Hello students, welcome to EPG Pathshala. I am Dr. Hidayatullah from Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today we are going to talk on model protein structure validation tool from paper bioinformatics. After completing this model, you should be able to understand about the possible errors, that means possible errors in the structure prediction method. Then what is the need to do a structure validation? Then we'll talk about the critical assessment of techniques for protein structure prediction. This is the community which is being developed to develop programs for model prediction. Then types of error which has been observed in the structure models. Then we also discuss about the structure accuracy, how to judge a model whether they are structurally validated or it is the model predicted is very accurate. We also talk about the validation programs to assess the quality of the predicted structures. So we will start this model with some basic questions in mind, because any structure which you are going to predict about it has to be related to the function of the protein. So we can't always believe on programs because they always mislead or often mislead. Sometimes they provide wrong observation also. So there is a difference between the mathematical calculations and biological interpretations. So the mathematical statistics should be correlated to the biological functions, because computers don't do biology, they do mathematical sum. Hence the researcher should interpret the structure with respect to the biological function rather than the statistical significance. So structure prediction becomes complicated if we don't know any information about the template and the query. So you should need to know about the sequence identity, the active site amino acids, the hydrophobic patches, hydrophobic fold, and all those information you should know before you do a structure prediction. If you have more information about the template and the protein whose structure has to be predicted, then we can avoid many possible errors during the structure prediction. The most important criteria we should follow is that the error becomes less dangerous when you know about them during structure predictions, so that we can correct them and predict a good model which can be related to the function of the protein. So in this case, there are many errors which we need to check before we select a PDB structure as a template for the structure prediction, such as technical error, whether it can be crystal specific error, or during data collections they must have done incomplete data, or it's an NMR specific error where the certain regions are not being defined properly, or during structure prediction the fold assignment has not been done properly. These are the possible errors. Many more errors we'll discuss in this model. Before you do a structure prediction, it is always better to go through the validation report of a template structure in the RCSB. So in the left hand side you see a figure where you see a circle is been highlighted in yellow which shows the full report. This full report has the information about the overall quality of the structure, sequence composition, what are the experimental error which is being available during the structure prediction, then standard geometry of each amino acid residues like dihedral angle, bond length, all those informations, all the errors are being reported in this full report. The clashes which are observed, the van der Waals clashes, hydrogen bond clashes, solvent and ligand clashes, all those information will be available. Then there are also information about the ligand and the geometry, how it is binding to the active site of the enzymes. So it helps us to know the possible errors in the model, and it also helps us to check whether the structure model can be used as a template for the structure prediction for our protein of interest. So first go through this report, get the informations, whether all these informations are right, or whether you have to look at the resolution of the protein structure also. That also plays an important role when you do a structure prediction. So first go through this full report of the validation report. Then based on the report you can judge whether this protein structure or model can be used as a template for a structure prediction method. So from the validation report, what we get it from the RCSB, you have in the left hand side there are Ramachandran plot, geometry, stereochemistry, R factor, correlation between experimental data and structure, resolution of data, and all those informations will be available. Ramachandran plot will tell you that how many amino acids are in the allowed regions or generously allowed region or additionally allowed region. Then the geometry and the stereochemistry basically tells you that whether the bond angles and bond lengths of the predicted structures are very within the range of validations or within the dictionary values. Then R factor, particularly in case of extracrystallography, the R factor tells you whether the predicted structure is as a lower error. Lower the R factor, better is the structure. So this also depends on the resolution of the data. Then correlations between the experimental data and the structure, that means how well the electron density matches with the model, where researcher or the interpretation done by a researcher to model the structure in the electron density map. All this information is available in this validation report. Apart from that, it gives the stereochemistry, overall stereochemistry quality, like planarity of the aromatic amino acids, then chirality, which relates to the side chain conformation of each amino acid, phi-psi angles. Apart from the dihedral angles which are related to the secondary structure, there are phi-psi angles related to the side chains also. Then chi angles will also be helpful in understanding the interactions, whether the rotamers is available for interactions, whether the rotamers is forming proper interaction with the side chains, all those informations. Then non-van der Waals contacts, all those informations are available. Then it also tells what are the errors which are unsatisfied, like unsatisfied donors, acceptors, what are the errors which are there in the structure. So once you read this validation report, you can assign whether this model can be used as a template for the protein structure prediction. The critical assessment of technique of structure prediction. This is a community which played an important role in boosting the computational based structure prediction methods. This community is consists of large number of groups. Their goal is to make softwares or server which can predict very good models. The major goal of this CAS community is to address the difficulty in protein structures. So the first meeting for this group was held in 1994, and every 2 years the meeting is being held. In this meeting, they discuss about the model predicted along with the experimental model, and they assess difficulty and how to improve the structure prediction servers. So they discuss about this one. So for more details about this CAS community, you can visit the website, which is www.predictcenter.org, to check recent meetings and target list and prediction, new programs which been released. All this information can be obtained from the website. The model I discussed about the CAS community. Although the CAS community helps in finding the difficulties in predicting the structure model, after predicting the model has to be validated. So the model quality has to be assessed by some programs, which is model quality assessment programs, which help in addressing and assessing the global or local accuracy of the predicted model. These model quality assessment programs have a several classification in assessing the protein models, like methods to evaluate the quality of the template and target sequence alignment, whether the sequence, the conserved region is been aligned properly, whether the catalytic amino acids has been aligned properly. Then it is also methods to evaluate stereochemical irregularities in the predicted models. It also has methods to integrate several features into the quality assessment by giving some score. So before you select a model for a model prediction, you need to predict the model by comparative modeling. There are two important information which are required. One is the target sequence should be in a detectable sequence similarity with the template structure. This will tell you whether the model which you are going to predict will have a good similarity or a prediction is right with respect to that of your protein of interest. So the computed alignment between the target sequence and the template structure should be accurate. So in the left hand side you see a graph which tells you the percentage of sequence identity and the percentage of structure overlap. If you see in the x-axis, you have percentage of sequence identity. So if you have 10% sequence identity, then you see the overlap between the structure predictor, it's very far. So as the sequence identity keeps on increasing with respect to that of structure model, you will see that error keeps on decreasing. If you see a cutoff there, 50%, 50% is the cutoff where you see more overlap between the structure predicted and the sequence alignment. So if you have fixed 50% sequence identity with the template structure, then the model which you are going to predict will have a very good sequence identity, structure homology, and the function which you are going to predict will be correlated beautifully. So the overall model accuracy depends on the degree of sequence similarity. Higher the similarity to the template, more accurate is the model. Although low accuracy models may also provide core structure and functional annotation, but it is always better to use high sequence identity model to predict your structure of your protein interest. So when you do a structure prediction for a protein structure, there are two major reasons where your predicted model or a homology model or a threading model to have error. One is the sequence identity between the query of the interest, that means your protein of interest, and the template model is low. If the sequence identity is low, if the alignment shows a very low sequence identity, then wrong incorporation of amino acid or residues may result in a packing problems where you have overall structure predicted in a wrong way. If your template structure has error, then it is obvious that the predicted model will also carry those errors. That is also need to be checked. Then hence it is essential to check the correctness of overall fold, structure, or errors in the localized region, stereochemical parameters like bond angle, bond length, geometry, basic geometry parameters like bond length, dihedral angles, backbone conformation, Ramachandran plot. All these informations can be obtained from the validation report at the RCSB website. So what are the major possible errors which can be observed in a predicted model? One is completely wrong. That means the wrong trace. That means the interpretation or the researcher has incorporated wrong trace. Instead of right-handed, left-handed helix is been incorporated like that. If there is a wrong fold, or instead of a helix he has made that as a loop, or instead of strand he has made that as he has in model that as a loop. These are the wrong trace. Then registry error. Instead of an alanine position he has put another amino acid cysteine. So if there is a wrong trace where the trace of the protein is not matching with the sequence order. Then there are partial error, like where you have incorrectly built loops, the loop has not been built properly, or wrong amino acids. Instead of glycine, arginine has been model. So this information creates error in the predictor models. So anything which related to the quality of the predicted model, it's related to the geometry and stereochemistry. So if you have a bad bond length, bond angle, or dihedral angles, it leads to the bad geometry. The overall stereochemistry of the protein structure becomes invaluable when you try to predict the function. So incorrectly positioned ligand will also be happen when if you don't predict the catalytic site properly when you do a structure prediction. So I was discussing about the types of protein structure errors in the protein structure model, like completely wrong, partial error, bad quality, all those information I was talking in the previous slide. So in this one we are talking about an example where we'll see how a wrong fold or incorrectly built model will lead to a wrong predictions. So here you see an example where you have a wrong fold incorrectly built model with a helix being modeled as a loop. So here in the figure you see the one which is highlighted by blue and it is blinking. It is a experimentally predicted model or a template structure which is being used. So here it is being predicted as loop. On the right side you see same sequence where you have predicted to be an helix. And more important thing is that you see in the left hand side the cysteine is no longer forming a disulfide bridge with the loop, whereas in the right side you see this is the correct fold where the disulfide bridge has to be formed between the two monomers of the protein. So if you take a template where the secondary structure is being predicted wrongly, then the model which you are going to predict will also be having error. So what are the possible errors? The error might be due to template model determined with poor experimental data, like it may be due to low resolution or due to the data incomplete data. So in case of X-ray crystallography, in NMR there might be peaks which not been resolved properly, so you might have this error. The error may be due to the template model where the researcher who is involved in solving the structure made wrong interpretation of the experimental data. For example, he must have predicted this to be loop rather than as a helix. If he has predicted helix properly, then your template model will be right. Then you can use that template model. The predicted structure might be also right. So the error might be due to the researcher's interpretation. The error might be due to wrong sequence alignment with the template model. Like when you do a sequence alignment between your template and the protein of interest, if you do wrong protein sequence alignment, then the possibility of shifting of the disulfide or the cysteine might have happened, where it leads to a different secondary structure prediction, which leads to this sort of difference in the fold. So this register error where the trace of protein is not matching the sequence order may be due to this following 4 or 3 reasons. So that's why it is always better to read the validation report from the protein data bank. So once you know what are the errors which has been there, then based on the error you can rectify the error. It's always better to know the error and so that we can rectify it's rather than not knowing the error. Apart from the wrong fold, the side chain packing is also an important aspect of structure prediction, where wrong incorporation of amino acid will lead to clash or improper fold in the protein model. So earlier you see example where the helix has been predicted as a loop. It might be due to the improper incorporation of amino acids, which lead to a loop conformation rather than a helix conformation. So that is due to the result of side chain packing. So the protein side chain packing always depends on the rotamers of the amino acid residue. So a protein side chain might have different rotamers. So protein side chain may have different conformation, may have different rotamers. They are mostly defined by the dihedral angles, or it might be due to the bond length, bond angle, which is relatively fixed. So most of the times the protein side chain conformations depends on the dihedral angles. That means it depends on whether it is lying on the helix or it's lying on the beta sheet or strand or loop. So the protein conformations or rotamers of the side chain will depend on the locations. Basically it depends on the dihedral angles of the side chain. So side chain conformation plays a major role when you do a structure prediction. So side chain packing error or to be rectified, because if the side chain has not been properly packed, then it is possible that the folding will lose when you do energy minimization during the structure prediction. So the rotamer conformations is also plays an important role in the structure predictions. So to define a rotamer, the dihedral angle of rotamers for a particular residue, it can vary from 0 degree to 360 degree. However, certain angles are energetically favorable, and these angles are called Gaussian plus, that is plus 60 degree, or Gaussian minus, which is minus 60 degree, or trans, which can be 180 degree. So the average Gaussian value from a finite number of possible dihedral angles are called as the favorable conformers or rotamers. These rotamers in a sense are a discretization of the dihedral angle in a space of amino acids. So these rotamer angles, these are generated in a library. These rotamer libraries are collected by selecting a unrelated protein structures. It can be anything, any protein structure which are higher resolution protein structure, because we are interested in knowing the dihedral angles for the amino acids, not on the protein structure. So you get rotamer libraries, different angles from the higher resolution structures. You create that rotamer library built on the conditions that backbone conformations where the phi psi angle should be in a space of 10 degree difference. These are called as a backbone dependent rotamer library. So we were discussing about the Gaussian 60 plus Gaussian minus 60 and 180 trans conformation of the side chain rotamers. So let's see an example. In this figure you see two examples. One is the histidine conformers and the tyrosine conformers. The figures shown below are all are energetically favor conformers of histidine and tyrosine amino acid residue based on the library developed from the database of high resolution crystal structures. So based on the crystal structures, the histidine 3 conformers from here. So tyrosine also conform 4 conformers. So but these conformers are ideal. It's not ideal for structure prediction. Which rotamer is ideal for this for the structure prediction depends on the environment where it is being introduced. So we see that histidine and tyrosine conformers are derived from the favorable conformers from the database of high resolution crystal structures. So the ideal conformer or rotamer suitable for the predictive model depends on the surrounding environment of the residues. So for example, if you see in the left side, you have tyrosine with 2 conformer 1, conformer 2. The conformer 1 is ideal, it is going to have an interaction with the aspartic acid, but it is too close to the aspartic acid, so the interactions are not right. So whereas in the conformer 2, you see the tyrosine is in an ideal distance where it can form hydrogen bond interaction with the aspartic acid. So conformer 2 is the ideal conformer rotamer for the tyrosine. So in example 2, where you have 3 and 4 conformers. So you see in the third conformer, if you use this conformer for the tyrosine, then it will have a clash with the histidine. Similarly for the conformer 4, if you use this conformer, then it will have a clash with the glutamic acid or aspartic acid, which is closed in the environment. So the ideal conformer for tyrosine is the rotamer number 2. Let's take an example for the histidine. Histidine, we show here 3 rotamers, 1, 2, 3. So rotamer 1, which is ideal, forming a salt bridge interaction between the aspartic acid and histidine, which is ideal. Whereas the rotamer 2 and rotamer 3, if you use those rotamers, then they are going to have a clash with the side chain, which is not ideal for the structure prediction. So we have to choose the rotamer 1 for the structure prediction. So this your program or the validation structure prediction server should be able to choose the conformers where it should have conformer 1 and it should be in the minimum energy conformation. All this prediction, all these parameters should be taken into account when you do the structure predictions. So conformers of each amino acid residues depends on the surrounding environment apart from the energetically favorable conformations. The major server which I will recommend is the SAVE server, which is a structure validation server. This server has all the programs which are required to validate your protein model. SAVES is well known as the structure validation server, which is available free to check errors in the structure models predicted by homology modeling or thread. So it is serves.mbu.ucla.edu. SAVE stands for the Structure Analysis and Verification Server. This is a expanded forms for the SAVES. This server is maintained by Molecular Biological Institute at University of California, Los Angeles. Let's see an example for a server where you have all the programs which are required to validate a structure predicted model. So SAVES is a server which stands for Structure Analysis and Verification Server. So it has many programs like ProCheck, WatchCheck, ERAT, Verify 3D, Prove, Crest1 Record Match, Ramachandran Plot, Well Model Viewer. So ProCheck basically tells you all the geometry, stereochemistry of the protein model, Ramachandran plot, all this information can be obtained by ProCheck. WatchCheck will tell you how well the stereochemistry of the model is been predicted, and it also tells about the solvent around the model which has been predicted. So this tells you the non-bounded interaction between different atom types. So what are the errors, what are the possible clashes which are obtained from the structure model? So non-bounded clashes, hydrogen bonded clashes, salt bridge clashes, all those informations will be displayed in the error plot. So on looking at the error plot, you can modify your model, or you can check those regions which has the error and try to rectify those errors. Then Verify 3D, it also tells about the assignment of the sequence with respect to that of a template structure. It also tells about whether the alpha helix, beta sheets, loops or polar contacts has been assigned properly. All this information can be obtained by verify 3D. Then prove is the one which tells about the active site or the cleft found at the models, all those informations. Then it also tells about the van der Waals contact, volumes, and all those stuffs can be obtained from prove. Then you have Ramachandran plot which tells about the amino acids lying in the secondary structure prediction, whether it's in alpha helix, beta sheets or loop, or it's in the disallowed region. All those informations can be obtained. So this save server has all the programs linked together, so you can take the model and you can upload into this server, and you can use all the programs to do simultaneously to validate your protein structure model. So let's take an example and do a project validation from the server. So you load your PDB or a model which you are built using some structure prediction server. You upload the model into the project, then you click run. Once you click run, it depends upon the protein size. It takes from 5 minutes to 20 minutes to validate a protein structure model. So once the validation has been done, it will give you a report. So here we are seeing two reports. One is the stereochemistry quality and the other is the files which are generated from the project. So it tells the coordinate file name, it tells what is the chain ID. Here we are not interested in the, if you are using the PDB, then the resolution will be reflected if you are using a crystal structure. Otherwise the resolution doesn't make sense if you use a structure model. Then it also tells about the side chain atoms swapping, whether the glutamic acid or aspartic acid needs side chain swapping for them to have good interactions. Then it also tells about the non-bonded interactions, calculation of bond length, bond angles. On the right side you see plots for everything, like for example Ramachandran plot, then all residues plot, individual plots, Kai plots, main chain parameter plots, side chain parameter plot, residue by residue plot, and all those informations. We will be discussing about each plot and how the results look like and what are the interpretation you can do from the project outputs. So these are the three plots which I am showing obtained from the project output. So if you upload the model into the project server in the server, and if you run project, so you have run the project and this is the output plot. So Ramachandran plot, here you see, as you know, every Ramachandran plot has an allowed region which is in the red, which is most favorably region. Then the yellow one is the one which tells you the generously allowed. Then the light yellow one is the one additionally allowed. Then the white region is the disallowed region. So you see the black dots which represents each amino acids. So the amino acids which are in the most favored region will lie on the red region. So there are also amino acids which lie on the generously allowed region. Then you see some of the dots which are lying in the outside, which are disallowed regions. So these amino acids we have to go and check whether it's lying in the alpha helix or a beta sheet or in the loop. If they are lying in the loop, then it's possible that due to the flexible nature or due to the template model which doesn't have the regions to be predicted, you might have some amino acids lying on the disallowed region. Generally most of the amino acids should be in the allowed region, but there are cases where you see amino acids lying in the disallowed region, mostly in the loop regions. So if they are lying in the loop regions, then it can be justified that these amino acids positions cannot be predicted. In general, you should have most of the amino acids lying in the most favored or additionally allowed regions. So it should have 100% in amino acids in the most favored region. And next plot is the Kai plots, which tells you the side chain conformation, basically rotamers of the each amino acid residue. You see here you have colored different plots for each amino acid. Totally in the structure which I have uploaded has a 15 arginines. Out of 15 arginines, how many are lying in the regions which are favored and which are not lying in the favor. All those informations will be obtained from the chi plots. So for each amino acid, like arginine, the number of amino acids, I have given only the amino acid for 9, but the plot, there are plots for all the amino acids. Just for an example, I have taken 6 amino acids and their plots. This is how the plots looks like. So the which are not lying in the ideal values will have a red color, rest are in the yellow, which are acceptable values. Then the right side we have a main chain parameter plot, which also gives for each individual amino acids. So each individual amino acids will have a main chain plot. So if you see a dot there, so this is the blue graded region, is the region where your ideally this amino acid should lie. This is the angles where it should lie. So you see a dot spot lying in the region, whereas on the right side you see the dot spot lying outside the region. So this is the disallowed region for the main chain parameters. So you need to modify or adjust or do energy minimization in for those amino acids so that it lies into the ideal parameter value. So each amino acid should lie within this range. If they are outside this range, then those regions need to be checked and the error need to be rectified. So let's see another two plots. One is the main chain bond length plot. So this tells you the bond length for the CN, CN, CO, CAC, all those bond lengths are been displayed. So the ones which are having error will have a shoot up bar like this. So minimum error, then it will be like you won't have any bar diagram. So in the, for example, CN, there's no error. Basically the regions are perfect. So there are some regions where the C11O has a high error. So these are the things which will be obtained from the project. So once you know that there is the bond length is not matching, you should go and check those amino acids, rectify those bond length, and do some energy minimization and upload the model in the project and check whether the bond length has been rectified. So on the right side you see distorted geometry for the aromatic amino acids or the side chains. So all the amino acids should have the planarity. Planarity is very important for them to show good interactions or to predict any interaction between two amino acids. So here you see the planarity. Basically the aromatic amino acids are more important, where the planarity plays very important with the stacking interactions. So ideal, the project will list you the amino acids which are not having the planarity. So these are the amino acids which are listed, and each amino acid residue number and how far it is deviated from the ideal value. Everything will be, all the information will be available in the project. So you go residue by residue, rectify the amino acid, do energy minimization or do planarity check of this amino acid, rectify this error, predict the model perfectly, and again you run a project and check. In many predicted models, it is often observed that the planarity, bond length, bond angle get compromised when the model is used for the molecular dynamics simulations. So it is always better to rectify the error before you do any further MD simulation studies. So this is the overall summary of the project output. So in the project output, if you are using a crystal structure to run a project, then you will be seeing the resolution, that is 1.5 Armstrong. If you're using model, then this doesn't make any sense to look at the resolution. Then it tells about how many amino acids is being used for the project summary. So it tells the Ramachandran plot. So you have 85% in core, 14.1 in allowed, and 0.9% in the gentlest. Then there is no amino acid in the disallowed region. So this is perfect for you to tell that all the amino acids are in the Ramachandran plot favored region. Then it also tells about the chi plots. So in this chi plots, it has taken 149 chi plots. Out of that, some of them having worse, some of them are not having worse. Whereas in case of main chain parameters, 5 amino acids are better, one is worst case, which we have seen in the plot also, that one dot lying outside the blue region. So that is the worst case. Then you have deviations from the bond angle, bond length, the hedral angles. So maximum deviation which is observed in the protein model is 9.2. So lower the deviation, better is the model. Then you see bad contact 1, which is usually good, but ideally there should be 0. So if it could be rectifiable, then you should be able to rectify this also. Then bond angle deviations, dihedral angle deviations. Apart from that, there's a factor called G factor, geometry factor. This geometry factor, if the overall geometry factor is below minus 0.5, I mean higher the more towards the positive, better is the geometry factor. So if it is below minus 0.5, then it is not a good model. So it should be more towards the positive. This one you see overall geometry factor is minus 0.26, which is better compared to that of minus 0.5. So like this you get a summary of overall amino acids in the predicted model. So based on this summary, you can either rectify or check the model. Then once this parameters looks good, then whatever structure prediction or whatever function you're trying to predict using the model will be validated or will be having a biological significance. This is the other program, which is error plot. So error plot, basically error function is based on the statistics of non-bounded atom interactions in the reported text structure in comparison to the database of reliable high resolution structure. So in the left side you see a plot where you see the error value, then the residue, so from 20 to 180 amino acids. So the ones which are having a higher bar is the ones which are having the very high error. Lower the bar value, better is the good model. So the ones which are in the dark bar regions are having very high errors. So these regions need to be, these amino acids need to be fixed, whether it has to be energy minimized or the planarity is not good. So or the conformers or the rotamers which are predicted in the model are not good, which is having a clash with the side chains, other side chains. So this region need to be fixed in the predicted model. So this error plot tells you the van der Waals interaction contacts or non-bonded atom interactions clashes. So this need to be removed. So if you have a good high resolution structure model, then you structure and you take that high resolution structure and for model prediction, then generally the overall error factor will be lower in case of error plot. But if you use a low resolution structure from 2.5 to 3 angstrom, then the overall quality factor will be low. So in the error plot they also mentioned that low resolution structure will always have an error quality around 90 or something percent. So it depends on what structure you are taking as a template. So regions of the structure that can be rejected at 95% confidence levels are shown in the dark one, dark gray or black. Then 5% of the good protein structure is expected to have a error above this level. So most of the structure which is having error more above 95 are to be rejected, or you need to look at the structure. So regions that can be rejected or it has to be corrected is above 99% level. So based on the available high resolution crystal structure, this error plus has been designed for your model. So anything above 99% or 95% has to be reduced to 80% or 85%. So this region has to be fixed so that the overall error quality factor goes lower. Previously we saw example for pro check individually and error plot for individual run. Now let's take an example where we'll run all the programs together for a particular model. So where you will be running pro check, you will be running what check, error plot, verify 3D, prove. Crystal information is depends on the crystal structure. If you are using crystal structure or PDB structure to validate, then Ramachandran plot, then model viewer. So if you use all the, we can also run all the programs together for the structure. So here you see a color code which is green, yellow, then red. So green, which means good, satisfactory, then the yellow one is the one warning or inspection is suggested, then one with red is the one which tells that there are a lot of errors observed in the structure. So basically you upload the PDB file, or you can give the PDB code if you want to look at the parameters for the template structure, then you can give the PDB code also. So give the PDB file or PDB code, then run program on this file. So where here you see that please add this number, because this is to check whether the researcher is doing or any hacking is going on. So that's why this 9 plus 8 you see that number. So you upload your PDB or give the PDB ID and run all the programs on this file. Generally it takes half an hour to 45 minutes to run all the programs, and each one will produce a plot which will discuss in the later slides. So once you run the save program, the left hand side you see summary of all the plots like crest1, true, error, verify 3D, pro check and what check. Each one has a model. If you click that crest, it will give the output of the crest program. Pro, if you click pro, it will give the output of the pro program. So now the output which we are seeing is the error output. So I click the error output, and in the left hand side you see near the error plot you see some files which is in the JPEG format or PSC format or PDF file. So if you click, it will be displayed on the same window. So you see the error plot of the model, which you see the bars. As I mentioned earlier, the anything above 99%, which is confidence level is very low, this region has to be fixed. Generally the error should be less than that. So and if you have lower bars means your van der Waals contact regions are very good. That means there are no clashes in the predicted model. Now let's see the project output. So I clicked the menu project and I see the overall summary of the project results. So it gives in three format. One is the PostScript format, then the PDF file or the JPG. If you click each individual file, it will give all the information. Like if you click the Ramachandran plot, it will give the plot of all the amino acids. Or if you want to do a plot for individual amino acids, then the individual amino acid plot is also there. Then Kai plot is also there. Main chain parameter plots, side chain parameter plots, residue parameters plots, all those plots are available. So it is very important to go through the each PDF file, look at what are the errors been observed in the model, whether these errors can be rectifiable, or you need to do some energy minimization to rectify these errors. So you should do that one and run the program again. So it also gives the overall summary of the plot which we saw in earlier project output summary. Similar summary is also there. So if we click that PDF file or PostScript file, it gives you the all overall summary of the geometry file. Next, let's see about the verify 3D output. So if we click the menu of verify 3D, it gives again the plot. So verify 3D output contains 3D 1D profile of the protein model. This 3D 1D profile describes the residue environments from the three-dimensional structure in the one-dimensional string. How the amino acids are arranged. So it describes whether the area of the side chain of amino acid residues is buried inside the protein. It also tells whether it is exposed. It also describes of the fraction of side chain area exposed to the polar solvent, or it is exposed to the solvent, or the side chains are inside the protein, whether hydrophobic patch is there, hydrophilic patch. All this information can be obtained in the 3D 1D. The 3D profile is generated by the position dependent comparison matrix, which illustrate all the residues in the particular model are oriented in a proper residue environment. That means the rotamers which are assigned in the models are in a proper environment, whether they are having good interactions. Like for example, the hydrophobic amino acids generally should be away from the solvent. So whether these hydrophobic amino acids are buried inside the protein and they are properly guarded by the hydrophobic environment, all those informations can be obtained from the verified 3D plot. So I was discussing more on the save server. Apart from the save server, there are many other structure validation programs available on the website. So you see JCSG validation report, check, which is a separate program, or you can run through saves also. Then PDB-E analysis, which is also helpful in analyzing the PDB structures which are available in the RCSB or PDB-E data bank. Then project, which can be run along with the saves or separately. What if, solve, prove, Anulia, iterate, verify 3D, check. Either you can run individually or you can run from the save programs where it has project, what if, iterate, verify 3D. All this can be run from the saves. But from that you can run individually also. So students, let's now summarize what we have learned in this model. We have learned like what are the possible errors which can be observed in the protein structure and what are the programs which can be used to rectify the error. Because the structure relates to the function of the protein. Hence, the predictor structure should be error free so that the function, whatever function you are predicting, should not become meaningless. The structure can be predicted through homology modeling, threading or ab initio method, but the model should be error free because the essential geometries like bond length, bond angle, dihedral angle should be error free with minimum tolerable error, so that the whatever prediction you are doing should be have a meaningful biological function. So we should always use template structure which has no global error. That means it should not have a wrong fold where instead of helix you have a loop. So those global error should not be there, and it should have a minimum local error. That means there can be any experimental technique you take, there will be error will be there, but the error should be very local, which is very minimum. So we should always use structure validation. We should always do structure validation before using a predicting structure, and also you should validate the predicted model. That will help you to understand the biological functions. And I discussed more about the saves program, which is a very useful server which contains almost all the programs tools which are related to the structure validations like project, PDB sum, what if, and other programs which can be very helpful in validating your predicted model. So I will wish you good luck in doing structure validation, which is an interesting topic in the bioinformatics, to do structure validations and use this server to validate your protein structure model. Thank you for listening this model.